revision for NEET 2026. Okay, so I know that there is a lot of anxiety, lot of confusions and so much that's coming along your way. But today I'm here exclusively to help in making a better strategy, in making a better mindset, and making sure that you all are going to do the NEET 2026 to your best potential. Okay, so we are going to target a 360 on 360 in biology. Like I said, that is going to be our primary strategy. All right, so today let's start our class. But before that, where is the energy in my class, and where are my amazing future doctors, everybody? And remember, whatever I'm going to teach today, every single thing I'm going to help you, okay, in making your own notes. Okay, so what I generally call as active recall. What is active recall? When you know nothing, or when you're starting to revise from scratch, you write down every possible thing that is there in your memory. Okay, so then what happens is you utilize all of these small pointers, and you revise it just before the exam. Okay, so that's how you do an active recall procedure. So that's exactly how I'm going to take today's class. So everything that I'm writing, everything that I'm doing, you will be getting my handwritten notes in my Telegram group. So all you have to do is the link is pasted there in the chat box as well as in the description box. So just click on the link, join the channel, so that you will be able to download all your notes from there. Okay, now but let me hear from you. So we have Meera here. Great to see you, Meera. What about the others? Okay, so Meera, do bring in your friends as well. Okay, so that will be great to see all of you in class. Alright, so before I start with the cell chapter, let me ask you, which is the most difficult chapter you think is in biology? Which one do you think is the most difficult chapter? Ma'am, this one chapter, ayo, I don't want to get over it only. I don't know how to get over it only. I don't want to see this particular chapter in my exam. Which is that one chapter? Let me hear from you. Yes, everyone? Which is that one chapter? Which is that one chapter? I want all of y'all commenting for me what that one chapter is. Okay, and I'm going to make sure that I'm just not asking this for a general information. Okay, so I have enough general knowledge. I don't need to know that from you. But what I need to know from you is where you are finding the difficulty. Now, whichever you're finding the difficulty, I'm going to be taking it in one of the days. Okay, and I will make sure that when you finish this class and go, this chapter is going to be super duper easy. There's going to be no way you're going to rethink or re-question your understanding. Okay, so all your doubts will be cleared here. All that you need to know will be cleared here, and everything will be done for you. Is that okay? So that's the energy I need in class. So once the class is over, I want all of you to comment, yes ma'am, I'm good to go in this chapter. Or if you have any doubts, I want you all to comment it as soon as the class is over, because that will help me to discuss these doubts. Okay, or if later you're watching the video and you have any doubts, I want you all to comment it, so that in the next class I'll be taking through the doubts first. Okay, I'll be taking your feedback first for what is the difficult part, any explanations needed and everything, and then we will go into the next chapter. Okay, so that's what we will follow in our class. Is that okay, everybody? Is that okay, everybody? Yes. So that's the ultimate spirit that I need from all of you. Okay, so I'm just waiting for a couple more minutes so that everybody can join in, and then we are good to start. Okay, so where is the difficult chart? Okay, Meera is saying, ma'am, animal kingdom is the most difficult. Okay, animal kingdom can be done, Meera, don't worry. I'll give you best mnemonics. I'll give you the best things so that you will be able to crack it. Okay, so apart from that, remember that clearing NEET is not a very difficult task and all. Okay, it is just that you are in a competition, okay, with so many lakhs and lakhs of students. That is where, you know, everybody finds a difficulty and everybody thinks that, you know, it is such a difficult exam. All this is only because of the kind of because of the kind of competition that you have. Otherwise, syllabus wise, or, you know, understanding wise, remember that it's very, very easy. But the problem is, let's say you understood 70% of the chapter. Great. Somebody else understood 75% of the chapter. Somebody else understood 85% of the chapter. So the more better your understanding is, the better your rank and your score will be. Okay, so that is what we're going to channelize, and that is the only thing that we're going to focus on. Okay, so let's start our class, shall we? Yes, everybody. Okay, great. All right, so let's begin our entire class. So first, when you see cell, the unit of life, what is the cell? Rohit, Meera, what is meant by a cell? So cell is your structural as well as the functional unit of life. What does that mean? Structural, because we are made up of cells, right? And functionals, because all the activities in our body is all performed by the cells. Now when we look at cells, right, we can say there is two different kinds of categories when we look at cells. So first is going to be the unicellular cells. Means what? Only one cell is going to do both these functions, okay, or rather all of the functions, all the activities, digestion, respiration, okay, everything is going to be done by the same cell. That's what we call as a unicellular cell. But when I say multicellular cell, means there are many cells who are doing different kinds of activities. Okay, so that means every task that we have in the body is performed by different groups of cells, by different kinds of cells. Clear? Is that clear, everybody? Okay, so now when we look at unicellular and multicellular cells, we can further categorize them by calling out the different kinds of cells based on the structure. Okay, now what are these structures? Now let's say that we have different kinds of spaces, right? We have an office, we have a school, we have a building, we have a hut, we have so many different kinds of buildings that we can find in the city. So like that, when you look at the arrangement and organization of cells, we can consider them to be two important types, which is the prokaryotic and the eukaryotic. But before going into that, okay, before going into prokaryotes and eukaryotes, we have to understand something called as the cell theory. Okay, now what do I mean by cell theory? Now when you look at history, it's not like we always knew cells existed, correct? So we had the microscope being found first, okay, then from the microscope we had scientists who were looking at it and saying, okay, this kind of cell looks like this, that kind of cells looks like this, and started to analyze the organisms, analyze the cells. So here we have an important scientist, okay, called as Schleiden. Now Schleiden was a scientist who simply said, okay, I looked at plants. Plants are made up of cells. Okay, that's all he said. Now then we had another scientist called Schwann. Schwann said animals are made up of cells. Same. Okay. Then we had another person, another scientist called Virchow. Now Virchow said, now there are cells, but where are these cells coming from? They come from, okay, cells arise from pre-existing cells. This is what he said. Now together, what do we call the cell theory? It is the fact that all organisms are made up of cells. First thing. Okay, and then like Virchow said, the same statement, cells arise from pre-existing cells. Okay, this is the ultimate cell theory. Okay, so we understood that cells are basically making the all plants, animals, and the entire framework of the individual, and they only arise from pre-existing cells. It's not like from somewhere suddenly you will get a set of cells appearing, right? So these cells arise from pre-existing cells. Is that clear to all of you? Now, when you look at the overview of a cell, okay, now what a cell will basically contain is it will have a plasma membrane. Okay, so the plasma membrane is like the security gate. Okay, we have a plasma membrane, and then we have a nucleus. Who is the nucleus? Nucleus is basically the head. Okay, somebody who controls the entire space. And what is the space that we are in? That is nothing but the cytoplasm. Okay, so we have plasma membrane, we have nucleus, and we have the cytoplasm. All of this together make up the cell. But now when we look at plant cell, animal cell, prokaryotic cell, eukaryotic cell, we might have some plus minus aspects, but these three are definitely non-negotiables that you will see in all the cells possible. Okay, now we will talk about the different categories of cells that we have. So the first cell, what did I say? It is called as the prokaryotic cell. And the other one is called as the eukaryotic cell. Now when I say prokaryotic and eukaryotic, pro means primitive. Okay, so imagine the kind of phones that you have, right? We have basic prokaryotic cells will be something like the old Nokia phones. Okay, so the Nokia button phones, if you all remember, maybe your parents can help you to understand how it was back then. We didn't have all the touch screen and all the fancy things in the smartphone, so we had like a very basic button phone. So this prokaryotic cell is pretty much like that. All your bacteria, okay, all your cyanobacteria, okay, all bacteria, mycoplasma, everything will come under this prokaryote category. Okay, whereas when you look at eukaryotes, there you will see all the other animal cells, plant cells, all the little bit advanced kind of cells. Even unicellular cells can come under eukaryotes, but these are something that have a little more advanced sort of a cell structure. Now when you look at prokaryotic cells, what does their cell look like? Fundamentally, they have a cell envelope. Okay, so this is like covering, okay, covering the entire cell. We have an envelope, and then we have a cytoplasm, which is basically the jelly-like space that is present in there. Then they have something called as the nucleoid. Okay, nucleoid is basically the nucleus region, but here they don't have any separate room, separate compartment for the head to come and sit, or the nucleus to come and sit. So there is no defined nucleus, but all the genetic material and everything controlling the cell will be in a region that we call as a nucleoid. Then we have what we call as the ribosome. So ribosome will make sure that all the protein synthesis is happening. Okay, then we have something called as inclusions or inclusive bodies. Okay, so I'll tell you what each one of these are. Okay, so firstly, starting with the cell envelope. Okay, when you look at the cell envelope, we have mainly three layers that will make up this cell envelope. So firstly, we have the glycocalyx layer. Okay, so the glycocalyx is basically for the protection of the entire cell, for protection. Then we have a cell wall. Now what the cell wall is going to do? Give basic shape and support. That's all. Simple framework function only this has. Now next, what do we have? We have a plasma membrane. Okay, so plasma membrane has a very specific property called as selective permeability. Okay, so selective permeability means everybody is not allowed. Only some people are allowed, and I will decide who should come and who should not come. End of it. Okay, now apart from that, there are structures called as the mesosome. Okay, so mesosome helps in the infolding of the membranes, okay, which is very advanced kind of function that happen during respiration or in DNA replication and other things. Then apart from that, we might have other chromatophores that we say. Okay, these are colored pigments. Specifically, we can see this in photosynthetic bacteria like cyanobacteria. So they help in photosynthesis. So these things are possible. Now apart from the cell envelope, okay, on top of these three layers, we can also have some surface structures, okay, which include flagella. Okay, we can have pili. Okay, we have pili, and then we can have cilia. So all of these things can appear in the surface as well. Clear, everybody? Now coming to the nucleoid region. Okay, so nucleoid region is where we will find the DNA. Okay, the major DNA in generally prokaryotics is often a single cellular DNA that you will find. Okay, is that clear, everybody? Clear to all of you? Alright, so this DNA is generally held in the nucleoid region with the help of certain proteins, okay, and that will give a little bit of stability so that even if they don't have a compartment, it's not like everybody is lying everywhere. Okay, so they still have a space, they still have a region where they will be sitting and doing their work. So that's what a nucleoid is. Clear? Now apart from that, the ribosomes, okay, so when you look at ribosomes in prokaryotes, these are what we call the 70S type. So they have one small subunit, they have one large subunit, so that is nothing but a 30 plus 50. Okay, so 30S plus 50S we say, so that makes up a 70S ribosome. Okay, now apart from that, I said there is something called inclusion bodies. Right, now these inclusion bodies are nothing but storage. Okay, so if you have a small house, then you have one cupboard, or you have one part of the house or one room in the house where all the storage will go. It's like, okay, I'll take it later, I'll put it inside this room, kind of a thing. So for all the storage purposes, they have something called as the inclusion bodies. Okay, so what do we have? We have the cell envelope, we have the genetic material of the nucleoid, okay, region, and then we have the ribosomes, and then we have the inclusion bodies. Now, in the nucleoid, remember that most prokaryotes have something called as the plasmids, okay, in prokaryotes, which is nothing but an autosomally replicating, means it will replicate on its own, and it's a self-replicating, okay, self-replicating extra chromosomal piece of DNA. Okay, means these are not belonging to the core genome of the cell, but these have a little bit of these are positioned outside of the entire genomic bulk. Understood, everybody? Now coming to the prokaryotes. Ma'am, eukaryotes is over. Yes, it is over. Okay, so very simple prokaryotes are. Okay, very primitive they are. Very simple, very basic. This all. If this much is there means okay, prokaryotes are happy. Now apart from that, they do have some small organelles, but they do not have any double membrane super functioning proper organelles in their system. Okay, now when you look at the eukaryotes, okay, now eukaryotes include protists, plants, animals, fungi. Everybody will come under this umbrella of eukaryotic organisms. Okay, now when we say eukaryotic organisms, these are these are structures that have proper membrane bound organelles. Okay, so these are proper membrane bound organelles. Apart from that, they have a true nucleus. Okay, they have a true compartment that will take care of the genetic material, and they're not mixing with everybody else. They have a separate room, separate compartment, and they also have cytoskeleton. Okay, they have cytoskeletal proper features that are present in their system. Okay, so all of these are present in the eukaryotic cells. Now, as is, we can split the eukaryotic organisms into plant and animal, okay, which have slight differences. Okay, so when I say plants and animals, only basic things I'm going to highlight for you. Okay, now when you look at a cell wall, definitely is present in plants, but it is not present in the animal cell. Okay, now we have some structures called as the plastids. These are plastids, we say leucoplasts, okay, or chloroplasts and other pigmented small pocket like structures in the cell. And these plastids are there in the plants, they are not there in the animals at all. Now there is a structure called as the centriole. Okay, now centrioles help in cell division, and these structures are present in the animals, but they are not at all present in the plants. Okay, now apart from that, structures like the vacuoles. Okay, the vacuoles are nothing but storage pockets. Okay, and these are pretty large in the case of plants, whereas they are sometimes tiny or sometimes absent also in the case of animals. Okay, everybody, this is the outline. Now when you look at the eukaryotic cell, the first person that you have on the outside is your cell membrane. Okay, or what we call as a plasma membrane. Okay, and for this we had a set of scientists. Now can you tell me who that scientist is? Yes, who is that scientist? Who is that scientist? Yes, Raghav, Rafa, Banu, Meera, Rohit. Yes, are you all clear so far? All well, Rafa, Banu, thank you so much. Okay, all right. Right, so, excuse me. So, when we look at the cell membrane, we had a set of scientists called Singer and Nicholson. Okay. So, what Singer and Nicholson did was they studied the cell membrane, and they told us that, look, the cell membrane is basically having two layers. Okay, so that's what we call as a lipid bilayer. Now how does the structure of a lipid look? They have a head and they have a tail, but the head is hydrophilic, means they love water. But the tail is hydrophobic, means they are scared of water. Now if I make a single layer, what will happen? Invariably one side has to face the water, this side or that side. But this fellow will get scared, so he cannot meet the water. So what nature has done is this exists as a bilayer. So what will happen? We have two layers wherein the outer side is made up of these hydrophilic heads, and the inside is made up of the hydrophobic tails. So in this setup, the tails are never going to meet the outside water, and so they are safe. Clear? So this kind of a lipid bilayer structure is present. Now over and above, these lipids provide a certain fluidity, means they can have some movement across them, and it is the proteins that are going to move laterally. Okay, so these proteins are going to move laterally, and there are two types of proteins that are majorly present. So we have an integral protein and we have a peripheral protein. So an integral protein is inside. Okay, so integral is inside the bilayer, so it is present somewhere like this, embedded inside. Okay, whereas peripheral proteins are something like, attached to the bilayer. Okay, so they are attached to the surface, only surface level proteins. Okay, now sometimes these proteins can bind with another phosphate molecule and form a phospholipid. Okay, sometimes bind with a glucose molecule and form a glucolipid, and so on. So like this, there can be many things that happen. Now this lipid bilayer, this cell membrane, is what I said is selectively permeable. Correct? So when I say selectively permeable, means there is some transport that is happening. Okay, I will not allow everybody inside, but I allow some people inside. I don't allow some people inside. So how do I allow? What are the transport mechanisms that are present in this particular system? If we see, firstly, we have diffusion. So diffusion means what? There is no energy, okay, and this follows what? A concentration gradient. From higher concentration to lower concentration, it will just flow, okay. I don't have to make any effort. Now when I say osmosis, that is also a kind of diffusion, but the only difference here, okay, is that here also there is no energy, but it is through a membrane, and through this membrane my solvent molecules will move, okay. Only water can move. Solute movement also can go from here to there, so it's not like a one way, both inside and out can happen. That's what we call as osmosis. And usually water, minerals, all of this go through osmosis. Clear, everybody? Then we have something called as facilitated transport. What is facilitated transport? Where somebody is going to help me. Okay, when I say facilitated transport, it means somebody is going to help me, right? So in helping, I can either help and go perform diffusion. It's called facilitated diffusion. Or anything, any molecule can just help me to transport this many molecules from here to there through a certain molecule, through a certain protein, or somebody who is in between and helping me. So for this also I require no energy. But we have something called active transport, where this will be first of all against the gradient. Okay, whatever higher to lower concentration we have, it will be against the gradient, and this requires energy, without which I cannot perform this function. Clear, everybody? Got it? Arun, Prakash, Meera, Rafa Banu, all good? Is it clear to you? Okay, perfect. Now when we say, so far any questions? Any questions that I need to clear before we move ahead? No? Okay. Now when we look at a plant cell, okay, when we say a plant cell is present, we have a structure called as a cell wall. Okay, so over the plasma membrane, we have something called as a cell wall, and this basically will function for protection. Okay, it will give shape. Okay, and it will also prevent, it's basically protection. Okay, it will prevent any dehydration. All of this will be taken care of. Okay, and what it is made up of is different from each and every species. Okay, for example, in plants we have cell wall made up of cellulose. Okay, in fungi we have made up of chitin. Okay, apart from that we have algae who use certain minerals also additionally on top of these basic things. So these kind of things are there, and inside the cell wall itself we have some connections that can also happen. So that's called as a plasmodesmata. Remember that plasmodesmata are nothing but cell connections in the cell wall. Okay, clear? Now that the outside is over, when you come inside, we have a very, very peculiar something called as an endomembrane system. Okay, so endomembrane system is what? Let's understand. Now basic, okay, do you all purchase on Amazon, or do you all do Swiggy? We all order on Swiggy, correct? We all order on Swiggy. Yes, anybody who's not used Swiggy? Anybody who's not used Swiggy? Ma'am, never eaten from Swiggy, ma'am. No. Don't give me heart attack like that and all. Okay, so I'm pretty sure all of you would have used Swiggy at some point in your life, correct? So now when I take Swiggy and I place an order, what will happen? Somebody has to cook, okay? Somebody will cook, and they will, put it in the boxes, and they will give it to a delivery person. This delivery person, okay, will take this packet, he'll keep it, he'll pack it, he'll do everything, and he will give it to the consumer. And then what you will do, you will either take it and keep it in your fridge, or you will eat it, or you will throw it. You will do something, correct? Now when you look at the cell, an endomembrane system does the same Swiggy business but slightly different. So whom do we have first? We have the endoplasmic reticulum. Okay, so endoplasmic reticulum is who? Is one person who is actually going to do the synthesis. Okay, then second, whom do we have? We have Golgi. Okay, all the Golgi apparatus, or we can also say Golgi complex. However, now this Golgi body is going to take care of the packaging and shipping. Okay, delivery to your house. Then what do we have? Then apart from this, we have the vacuoles. So vacuoles are nothing but for storage. So you take it and put it in your fridge. Or what you can do, you can eat them and you can digest them. So for that you have lysosomes to digest or discard, whatever. Okay, this is how it will look. So how exactly the system works? Okay, let's take each organelle one by one. Okay, and let us understand. So firstly, when I say ER, endoplasmic reticulum, we have two types. Okay, we have the rough endoplasmic reticulum. He will majorly have, so it's basically kind of like this, okay, thrown into folds like this, and he will have some rough structure on top of him. That is why we call it as a rough endoplasmic reticulum, because he is having the ribosomes. And what do ribosomes do? They do protein synthesis. So his primary function is to make proteins. Okay, protein synthesis. Then we have something called the smooth endoplasmic reticulum. Now what the smooth endoplasmic reticulum will do? This fellow has no ribosomes. Okay, so that means he's a smooth structure that is present, right? Now this smooth structure basically takes care of the lipid synthesis. Okay, this is what the endoplasmic reticulum looks like. Done. Now synthesis is over. Now whom am I going to give this to? I am going to give it to the Golgi. Now how my Golgi is going to look is, he's going to look something like this. Okay, with some proteins over here, over here, over here, over here. This is how his structure will look. Now this one side, which is basically convex in nature, is called the cis face. And this side, which is little concave in nature, this is convex. And whatever is concave here, we call as the trans face. Okay, so this is the receiving end. This is the shipping end. Okay, so now once the endoplasmic reticulum is giving protein or lipid, whatever it may be, I will receive it. I will nicely package it. Okay, I will understand whom I should give this to. Okay, I will make the full plan of who I'm going to give this to, and I will transport it from here. So whatever proteins are needed, whatever packaging is needed, everything I will take care of, and I will give it to the next system. Okay, so it's like I will make some small pockets with lipids or proteins or whatever, and this will be given to the cell. Now who all is using this? Everybody is using this. No, everybody needs protein, everybody needs lipids and everything in this thing. Correct? Agreed? Arun Prakash? Yes, Arun Prakash, don't worry. Yes, all sessions I'm going to do. Okay, sorry, all sessions I'm going to do. So don't worry at all, everything will be taken care of. Okay, hope my screen is, I'm not, yeah, okay, everything I'll be taking care of, don't worry. All right, and yes, plant physiology will be taken care, biotechnology will be taken care, everything will be taken care, don't worry. Okay, so do tell your friends also. Okay, I'm only targeting 360 on 360 in biology. Okay, and I know exactly what you need to do in order to get it right. Okay, so let's keep the energy up high. So far endoplasmic reticulum giving to the Golgi, everybody is clear? No doubt so far? Yes, all good? Okay, now what happens? Golgi has given. So all the other organelles who are present, no, they will all take it. Now endoplasmic reticulum will sometimes help in making the membrane also, detoxifying also. So all this it will take care of. Okay, so once the Golgi apparatus has given the proteins, has shipped the proteins, now what will happen? Everybody is using whatever they have to use, and then they are coming to the lysosome. So lysosome is basically you who is digesting the food. Okay, but in this case, what is going to do? All the other proteins who are used up in the cell. Okay, so Swiggy example I am giving it for you, but think of it as a cell. Once all the processes are over, okay, what it has is, first of all, in a eukaryote, I said we have a double membrane structure, but here lysosome is a single membrane organelle. Okay, so the single membrane organelle is going to contain what? Contains hydrolytic enzymes. Okay, so these hydrolytic enzymes are going to take care of what? Digestion of what? All whatever is remaining, all the biomolecules, whichever I am done, everybody will take care. Okay, so I'm going to have acids, I'm going to have people who are going to break down all of this thing for me. Okay, and then the last fellow part of this endomembrane system is my vacuoles. Okay, so my vacuoles are basically meant for storage. Okay, in plants, almost 90% of the cell is vacuole only. Okay, storage only is maximum. Okay, cell volume, maximum 90% is going to be taken care of the plant vacuole. Whereas in animals, it's extremely small or sometimes even absent. We don't know. Okay, so sometimes, I mean, not we don't know, some cells may have, or some people might be just absent. Okay, so they also have a membrane, and they will primarily work for storage as well as for osmoregulation. So they take care of what is the kind of ionic balance inside, okay, what is the pH, all of this my vacuoles are going to take care of. Clear, everybody? Any questions so far? Any of you all got? Okay, any questions so far? Yes? Arun Prakash? Yes, Pikachu, this is the first chapter I am taking. Okay, don't worry, this is the first chapter I am taking. So from today onwards, every day I'm going to be coming live and I'm going to be taking. So you can definitely be assured that I'll be giving you all these notes also. So you can clarify all your doubts in here. Okay, thus join the Telegram group, okay, that I have pasted in the chat box, as well as you will find it in the description box. So make sure to join so that you will get all of these notes over there. Okay, so you can refer to it and you can study. Okay, everybody? All right, clear. Now coming to the mitochondria. Okay, so this is not part of the endomembrane system. Okay, this is a separate organelle on its own, but it's a very, very, very important organelle because this is the powerhouse of the cell. Okay, and what does it make? It makes a molecule called as ATP, which is the currency, okay, which is the energy currency of the cell. So in order to use energy, I need this ATP. That is my energy. Now where am I getting this ATP? This mitochondria is going to make. How is it going to make? That is what you will understand in respiration, in photosynthesis and all of that. Okay, now when you look at the mitochondria, how does it look? It looks like a boat, okay, but that boat, I'm just going to open that boat and show. Okay, so from tomorrow, okay, I want all of you to keep your NCERT also next to you when I'm teaching, so that any diagrams I'm drawing, I can tell you, see, this is my rough diagram, but you can find this in so and so place. Okay, so I'll be able to tell you that. Okay, now firstly we have an outer membrane, outside membrane that is present. Okay, so my mitochondria became little oval because of space constraints, but please don't think mitochondria looks like this. Okay, so we're going to have an outer membrane, we are going to have an inner membrane also. Okay, now additionally inside we're going to have some folds, okay, which we will call as the cristae. Okay, inside the cristae, whatever space you see is called as the matrix. Outside the cristae, whatever space you see is the intercellular space. Okay, or you cannot say intercellular, you can say it as an intermembrane space. Okay, between the membrane you will have the space. Got it, everybody? Is that clear to all of you? Yes, this is how the structure of my mitochondria looks. Okay, and this mitochondria is the person who is involved in ATP production. Now this fellow has his own DNA. He doesn't have to wait for anybody to finish their work, give information, nothing. Okay, and how does it divide? It will divide by fission, just breaking into two. When cell division is happening, this will also break into two. The DNA will divide, and each person will get their own set of DNA when the cell division is taking place. Clear, everybody? Is that clear to all of you? Yes, so mitochondria, very, very, very important. Okay, now apart from the mitochondria, one more most important person in the cell is who? Is the plastid. Okay, so what are these plastids? We have three different major kind of plastids. Okay, first we have the chloroplast. So chloroplast, what it does? Majorly photosynthesis. Okay, so used for photosynthesis. Then apart from that, we have the chromoplast. So chromoplast will contain color, all kinds of color, pigments, everything chromoplasts will have. Then leucoplast. Leuco actually means white, okay, but these people are actually colorless and they are actually functioning for the storage purpose. Okay, now here the most important organelle you have to study is the chloroplast. Why? Because obviously meant for photosynthesis, okay, used in all of these processes. So 100% you will have to study about the chloroplast. Okay, so when I look at the structure of the chloroplast, okay, if you see the structure of a chloroplast, what do we have? We have the space called as the stroma, correct? And then we have some stacks. Okay, so stacks of what? Stacks that are present, and these stacks basically we call them as grana. Okay, so each individual person is called as a grana. Okay, and first of all, so understand that when you look at it, before even I go into the grana and the stroma, you have to understand that we have an outer membrane, inner membrane here also, because these people are also double membrane organelles. Okay, so you will have outer membrane and an inner membrane that is present. Okay, now here inside this, okay, inside this, what are we seeing? We are seeing that we have these structures called as the thylakoids. Okay, so we have small thing called as the grana, and these grana make up these stacks called as the thylakoids. Okay, and then we have some lamellae that will be connecting these different thylakoids. Okay, so it's kind of like arranged in stacks. I'm saying the opposite, am I not? So the thylakoid is the individual, these stacks is called as the granum. Okay, I think I said it the opposite, sorry guys. Okay, so these piles are called as the grana. Okay, so thylakoids and the granum is present, and then in between them we have the lamella that is present. Okay, clear, no? So I just kind of twisted that, yes. Okay, so I hope you're all not confused. So the individual is called as the grana, thylakoid. Together these stacks are called as the grana. So this grana and the thylakoids are present, and these thylakoids are the ones who will contain this chloroplast, and the kind of enzymes that are needed is present in the stroma, and together they will conduct the entire photosynthesis process. Okay, so the stroma contains the enzymes, remember, okay, and this grana is what will have the chlorophyll pigment to perform the photosynthesis. Okay, clear? Now coming to the ribosomes. Okay, I told you briefly, okay, when we were doing rough endoplasmic reticulum, but ribosomes are yet again a very, very important set of organelles. Okay, so these are nothing but protein factories. Okay, so protein synthesis factories. This is where the protein synthesis majorly will happen. Why ribosome is the cellular factory for synthesizing proteins, you will understand when we are doing molecular basis of inheritance. Clear everybody? So they have a large subunit and they have a small subunit. Okay. Now in prokaryotes, I told you it's a 70S ribosome that has 30S and a 50S subunit, whereas eukaryotes, okay, will have an 80S ribosome, and they have two subunits, which is 40S and a 60S. Okay, and this is basically, it is a sedimentation coefficient. Okay, so how they sediment is, if we just noting it, and it's actually called as the Svedberg's unit. Remember that. Okay, so this is what the entire breakup of the large and subunit is. How it functions to synthesize proteins, this you will obviously know when we are doing molecular basis of inheritance. Okay, now coming to the cytoskeleton. Okay, so cytoskeleton is what? These are nothing but filaments. These are kind of structures, okay, which are comprising of microfilaments, microtubules and all of that. Okay, and they are involved in support. They are involved in movement. Okay, so for support, for motility, okay, and also for the shape and support will come together, but okay, they provide the shape also to the different either. Okay, now what are the different cytoskeletal elements that we have? We have something called as cilia and we have the flagella. Okay, both of which are super important structures. Okay, now when we look at the cilia, okay, cilia and the flagella, majorly they contribute to movement. Okay, and they have something called as the 9+2 arrangement. Okay, if you see in your textbook also, there will be 9 filaments on the outside, 2 filaments on the inside. Okay, and they are slightly different in their support nature, and they will help to give the exact functioning of whatever this cilia or the flagella is working for. Understood? But they majorly participate for movement. Okay, then coming to the centrioles and the centrosome. Okay, now these are present only in the animal cells. Plants do not have this particular structure. Okay, so what they do is during cell division, okay, for the spindle formation we require this organelle. Clear everybody? That's it. Okay. Now coming to the most important, now we are talking about endomembrane system, then we spoke about the mitochondria, then we spoke about the what do I say, mitochondria, plastids, everything we have done. Now we have to talk about the head. Who is the head? He is nothing but the nucleus. Okay, so the nucleus also is a definite double membrane structure. Okay, so they have a clear nuclear envelope. Okay, and there are small tiny pores also that is present. Okay, these are nothing but small spaces where this envelope is missing. Okay, so these are nothing but nuclear pores. Now why these pores are important? Because obviously from the nucleus, once my processes like transcription is over, it has to come out for the translation and so on, right? So this is the fundamental place from which these exit entry and all this will happen. Then we have an internal region called the nucleolus, where the entire DNA is confined, okay, inside the nucleus also. Now this is nothing but the chromatin. Okay, if you do not understand clearly also, don't worry, because when we do molecular basis of inheritance, I'll tell you exactly what is DNA, what is chromatin fiber, how chromosomes are formed, everything we will take care there. But just remember that nucleus, of course, is the control center. Okay, he is the head, he is where all the processes are taking place, and it is where the genetic material is stored. Okay, everything is stored here and it is preserved here. Clear everybody? And here this chromatin condenses to what we call as a chromosome. Okay, so these chromosomes are something that we can say, nothing but condensed structure of this same chromatin fiber itself. Okay, so also remember that the cytoplasm in the nucleus we will call as the nucleoplasm, not cytoplasm. So nucleus, inside whatever cytoplasm is there, we will call as the nucleoplasm. Clear, everybody? Now coming to the chromosomes, like I said, they utilize some proteins. Okay, these proteins are called as histone proteins. I will explain them to you in detail in the next chapter, so I'm not really concerned too much about it. But when you look at the structure of a chromosome, okay, so they basically have a small constriction which we call as a centromere. Okay, this is the constriction between the two. Now, on the centromere, on both sides of the centromere, we have like this kind of lots of bolts that are attached. These we call as the kinetochores. Okay, CHO, sorry. Okay, kinetochores. Clear, everybody? So the centromere is the one that will hold both these parts of the chromosome, which we call as the chromatids. Okay, is that clear? Now, based on where this centromere is, we can divide the chromosomes into 4 different types. Okay, this is important, can be asked in your exam. So first we have a metacentric, where the centromere will be in the center. Okay, then we have something called the submetacentric. So submeta means little off, slightly off, okay, little above. Then we have a acrocentric. Okay, acrocentric means in the end. Okay, so like almost in the end, like that. Okay, so here metacentric is nothing but center, submeta is slightly off center, acrocentric is towards the end, and then last we have telocentric, which means not almost, end only. Okay, that is what we call as telocentric. Clear, everybody? Understood? So now coming to exam focus. Now if you attended the remaining part of the class or you don't attend also, I don't care, but exam focus definitely you will have to pay attention to. So irrespective of whether you watched my entire video or not, this particular part is super important for you. So exam focus is basically a list of topics, okay, a basic list of topics that I'm going to give you that can possibly come from the exam point of view. Okay, so you tell me as an examiner, what is this most common questions from this chapter going to be? Yes, Arun, Prakash, Raghav, Meera, Pikachu. Yes. Right. So like I said, irrespective of whether you watch the entire video or not, this exam focus is super important, because exactly these are the portions where questions will come in your NEET exam. So firstly, the different organelle functions. Okay, so what they do is super important. Okay, whether they give protection, whether they are involved in digestion, whether they generate energy, what they do is super important, can come as direct questions. Okay, then structural uniqueness. What do I mean by structural uniqueness? Now, for example, if I look at a mitochondria, it has a cristae, and then there is a matrix inside, and then there are 2 layers. Now, this is something you will not find elsewhere. Now, if you look at a plastid, if you look at a chloroplast, okay, you have thylakoids, grana, you have lamellae, stroma. So, these are pointers that they will, unique features that they will definitely ask for. Apart from there, lipid bilayer structure, spot on, Meera. Okay, the lipid bilayer and fluid mosaic model. Okay, who proposed it? Okay, so Singer and Nicholson. Remember the scientist's name also. I don't remember if I wrote it there, but definitely it will come. So remember that. Okay, apart from that, what you will have is differences. Okay, between animal and plant. they will not say differentiate between. It's not a subjective exam. But what is present in the animal that's not present in the plant? This you have to know, because directly they can ask you. They can ask you in statement-based questions. They can ask you in assertion reason. They can ask you in any kind of question. Okay, so animal and plant cell, prokaryotic, eukaryotic, very important. Okay, prokaryotic and eukaryotic difference. Okay, super important. And even the ribosomes, how they are different in each of them. All this you can just remember. Clear, everybody? Right? Apart from that, important scientists. Okay, this is the last priority, but still you should know. Like, for example, who proposed the cell theory? Who gave the lipid, fluid mosaic model of the lipid bilayer, okay, of the plasma membrane? All of this is super important. Okay? Clear, everybody? Yes? So, this exam focus part is super important. So, go through the video in case you have not, okay, and make sure that you revise this entire chapter. Now, just after this class, just think in your mind, okay, Shruthi Ma'am is already taking this class, and what I want you to do is just immediately go through your NCERT. Okay, class is over, okay, don't run away. Okay, so you have to just spend about 10 minutes, go through the entire chapter, read through that chapter, spend another 10 to 15 minutes, and this chapter is sorted. You don't have to look back at it again. Okay, and guess what, everybody? I have a very interesting tool, a very interesting feature that you can use for your practice. You can go on and on practicing, and you will get all your PYQ practice there. You can, understand how you are doing things, everything, but that is coming very soon. Okay, so I'm not going to tell you everything in the first class itself. So stay tuned. Do subscribe the channel if you still have not subscribed. Okay, like the video. Okay, and also join my Telegram group, because right as soon as I end this class, I'm going to share the notes into my Telegram group. Okay, so you'll get the notes there. So it will be super useful for all of you. So make sure that you are going to stay tuned. Every day I'll be seeing you guys. So tomorrow at 7 p.m. come back for a very interesting chapter, and we will do the same handwritten active recall technique. So just before the exam, let's say you're just traveling to your NEET center, or let's say you're just the previous night you're just looking for some resources, you just to revise whatever is important, then these notes are going to be super handy for you. Okay, everybody, so I'm really excited and really happy that all of you came here, and trust me, all of you are my responsibility. Okay, so any doubts you have, definitely do comment, definitely do subscribe, and I will see you tomorrow in class. Is that okay? Yes. So like I said, don't run away immediately. Make sure that you're going to do your this thing, NCERT reading once, okay, and then you can wind up and go for your next task. Okay, everybody, any questions? No? Are we done? Khalas. Okay, superb. So take care, everybody. Have a good night. Thank you so much. Okay, I hope you all enjoyed the class. Definitely do join the group for the notes, and definitely do subscribe and tune in for tomorrow's class. Okay, take care, all of you. I'll see you all tomorrow again. Bye bye.